Hello and welcome to the webinar on Nutrigenomics and hormone balance. My name's Emma Beswick and I'm the founder and director of Lifecode Gx, and we provide DNA testing, practitioner training and interpretations of DNA for health reasons. So we're going to talk today about briefly what DNA testing is, why and what and how, nutrigenetics and nutrigenomics, then a high-level overview of methylation, leading into the main topic, which is about hormone pathways and the genes and variants involved in that, then joining it all together and hopefully making sense of it. So why would someone do a genetic test? Firstly, out of curiosity, so direct-to-consumer tests like 23andMe or Ancestry really target consumers who might want to look at their ethnicity, ancestry, and maybe a little bit of health, but they tend to be focused on ancestry. And the health aspects can sometimes raise more questions than answers. The next 3 points are the ones that are of most interest to health professionals. So investigation is a very, very valid reason why, because genetic results can really help find the root cause of a problem when all else has failed, or if there's a family history of a problem. This can really help the investigation and find out why. So other reasons are prevention. So if you know that there is a risk in your family or you suspect a risk to yourself, you can actually test and find out someone's susceptibilities and vulnerabilities for certain conditions or certain causes of conditions. And this is particularly relevant for things that are chronic and long-term, that take a long time to evolve. By understanding risk early on, you can take proactive action before there are any symptoms. And then finally, optimization. So we often think of genetic testing as being about negative things and finding out about risk and it not being good and being quite depressing. But actually there is an upside, and it's a very strong upside, and that you can find out things about yourself that are good quite often, and that can help you optimize performance, whether that's physical or intellectual, and also help you live a long and healthier long life. So why? On the right is the functional medicine tree, which a lot of people will be familiar with. And you can see that genetic predisposition is right at the core of that. So the functional medicine practitioners have been using genetic testing for a long time as part of that big picture, part of the toolkit that they use to look at the whole person. And if you think about ATMs, so antecedents, triggers and mediators, genetics plays a big part in that as well. Clearly it's an antecedent, but it can also be a mediator. And what we're really seeking to do with genetics is to try and match a person's environment to their genetics. So to make sure the environment isn't disturbing or clashing with the genes, but rather supporting them. So that's the mediator aspect. And the word for looking at genes and the environment is epigenetics. So literally epi means over or on top of genetics, which is the environment overlapping and interacting with the genes. Okay, but the important thing here is to see genetics as part of a big picture alongside signs and symptoms, just part of that overall toolkit. How are genetic tests done? So firstly, we need a sample, and that can be blood, saliva or a cheek swab. So basically they all provide good quality samples, and obviously some are easier and less painful to collect than others. So we use cheek swabs. It takes 30 seconds to collect cells from the inside of someone's cheek, and it can be done at home, and it's completely painless, and we still get really, really good quality DNA from that. Then the DNA is sent to the lab, and it's extracted from the sample, and then it's sequenced. And what comes out of that sequence are a number of letters that you can see down there: A's, T's, C's and G's. So this is a bit more detail about that. You generally, in a DNA result, and this is taken from a 23andMe file, you would see an identifier, which is that RSID, which is the unique identifier for the place that you're interrogating on the genome. So you can see the ID, the chromosome, the position on the chromosome, and then most importantly, the two letters or alleles, that's the technical term, that are present at that location. And that can be any combination of A's, T's, C's and G's. They're the bases or the nucleotides that are present there. So we take the test result or the data and we compare it to our research database, which is curated research connecting genotypes, which is that allele combination. So connecting genotypes with phenotypes. So the phenotype is the result of that genotype. So how is it actually being displayed or shown as a health condition, for example? So we match that up and then we produce a interpretation of that information in the form of a report that explains the context, shows the results and explains the meaning of those results, together with protocols that might be appropriate for that result. And then finally, the application is you, the practitioner, using that information in clinic. Most genetic results tools and reports use colour coding, and typically a wild result or no variant would be positive. So 9 times out of 10, that's a positive version of a gene, or positive impact on a gene, and that's usually colour coded as green. If there is one allele change, so say the wild was AA, but someone has a T instead of one of those A's, that's called heterozygous. That usually indicates some negative effect. It can be positive, but roughly 9 times out of 10 it's a negative effect. So that's colour coded usually as amber. And if there are 2 changes at that point in the genome, that's called homozygous. So the example here is 2 T's instead of 2 A's, and that usually indicates more impact than an amber or heterozygous result would have, and usually that's colour coded red. In the Lifecode reports, we generally use colour coding to indicate the effect rather than just whether it's homozygous or not. So you can see where to focus on the effects that are negative, so you can try and mitigate those effects or risks and focus on them. Okay, nutrigenetics, nutrigenomics, what does that mean? So nutrigenetics is the effect of genetic variation on dietary response. So what is your response to a certain food group? And I've got some examples coming up. And nutrigenomics is the role of nutrients in gene expression. So that can be, how do certain nutrients affect gene expression? And an example of that is methylation, which we will come to in the presentation as well. So some nutrigenetics examples. LCT, which I'm going to go into more detail on, affects lactase persistence. So LCT is the lactase gene, and if it's switched on, it means that you will be able to continue to digest lactase. the VKORC1 gene is responsible for recycling or reactivating vitamin K. So vitamin K becomes deactivated as part of its blood clotting activity, and it needs to be recycled in order to still be useful to the body. There is a SNP on that gene that affects that recycling. So if it's disrupted, if that gene is less functional, it means that you can't recycle vitamin K, and you're more likely to be vitamin K deficient and more likely to bruise easily and bleed easily. Vitamin D, so the vitamin D receptor, VDR, if you have SNPs on that gene, that can affect your sensitivity to vitamin D, in that you are likely to be less sensitive. The so what there is, if you're less sensitive, you probably need to be at the higher range of vitamin D status in order to respond well to it. So that can mean for someone with those SNPs, they need to always keep an eye on their vitamin D levels. And then finally, in this list of examples, the HLA-DQA1 gene, SNPs on that can affect someone's response to gluten. So there is a specific variant that means gluten can trigger an autoimmune response resulting in celiac disease. So quite serious. Okay, so lactose in more detail, just one example we're using for this. So the lactase gene codes for the lactase enzyme. So that's often the case. The gene and the enzyme are often called the same, and lactase is responsible for breaking down lactose and turning it into galactose and glucose in this case. Quite unusually, so the 10% of the time, this SNP has a positive effect. So in the case of the lactase gene, the wild or ancestral version of the gene means that once a child is weaned, they become lactose intolerant. So the gene is effectively switched off at weaning, or when the child is about 2 or 3 years old. And so as adults, people become lactose intolerant, and that is historically what the position was. However, some people, and this is very, very closely related to ethnicity and geography, so Caucasians are more likely to have a variant on this gene, or that affects the gene expression and enables that to continue to be expressed even as adults, and so they can continue to digest lactose into adulthood. So in that case, they might have a ALL at that point in the genome. It doesn't matter whether they've got 1 or 2, so whether their genotype is GA or their genotype is AA, that enables that gene to continue working, and we can see that as a positive thing. So there it is as it may be presented in a report. So you can see the wild result is colour-coded red because that's perceived as being negative. A little bit of information and explanation about what that means and what the symptoms might be, and then the so what protocol. So yeah, the positive is the variant, and you should be able to continue drinking milk. Okay, so this is a new one. The FUT2 gene, basically a variant on this gene, which is referred to as W143X, determines whether someone is a secretor or not. So by secretor, we mean they secrete H and AB blood group antigens, and that includes on the GI mucosa. So the wild type in this case is the higher functioning gene, which is what we would expect from most wild types. And in this case, this also applies to if there is one variant. So the key thing here is to have a G allele. If you have the G allele, you will be a secretor, and that's the functioning version of the gene. If you have a double A variant at this location, so you're a homozygous variant, that's basically what the X refers to on the code, the W143X, because the impact of that variant is that it stops the code at that point. So say a gene is normally 50,000 bases long. In this case, just having that one variant, when that gene codes for the protein, it stops coding partway through when it hits that point in time. So you can imagine that that would have quite a big impact on the functioning of the gene or the enzyme if it's only a fraction as big as it should be. And that's the case in this situation. So in that case, the variant would result in a non-secretor type, which means those antigens aren't displayed. So that person would be a non-secretor, and that applies to about 20% of Caucasians. And it's a little bit different for East Asian people. And it is also worth knowing that we would look at a different SNP for people of different ethnicities. So that's detailed there. So it's just the research has shown that for Caucasians, we look in one place on that gene, and for East Asians, we look at a different place on that gene to determine the effect. Okay, so here's the colour coding again, and this is in the context of viral resistance and B12. So, you know, I was talking about good and bad, and I said that wild was good. Well, it depends what we're looking at. So when we look at FUT2, often it's looked at in the context of B12 as part of a methylation panel. And in this case, the secretor is not the beneficial type because that's associated with reduced B12 absorption and also more susceptibility to viral infection in the H. pylori. The variant or the non-secretor in that context has the benefit of being less susceptible to viral infection. In fact, they are resistant to Norwalk virus, the cruise ship virus, because that actually works by binding onto those antigens. And if they're not there, it doesn't have anything to bind to. So therefore, it can't infect you and it just goes straight out. So that's a benefit. And it's also associated with better B12 absorption. However, it's not all good for the non-secretor, and it's not all bad for the secretor, because FUT2 also has another impact, which is very different, but it affects the way that a mother produces breast milk. So basically, if the mother is a secretor, they are able to produce a substance called 2FL in the breast milk, and the impact of that 2FL on the baby is that it stimulates the growth of bifidobacterium in the baby's microbiome. So that's a very, very positive thing, I think, for a secretor. For non-secretors, unfortunately, they don't have that benefit. So non-secretor breast milk doesn't have 2FL in it, and so it's less protective. And so the research is that in infants fed by secretor mothers, on average, bifidobacteria were established earlier. So I think that's really interesting information to have, partly because we know what impacts that can have lifelong in terms of being able to to basically support that microbiome from the beginning. So that's just an introduction to a couple of genes and a couple of SNPs, and hopefully explaining the diversity and some key concepts. So now we're going to talk about methylation, and it starts to get a little bit more complicated. Okay, methylation. So what is it? It's the addition of a methyl group, quite simply the addition of a little chemical called CH3 as part of usually as part of a transformation of one substance to another. So during the transformation, something is methylated by adding CH3 to it. For example, methylfolate is a methylated form of folate. There are several steps to making it, but at the end of that step, we end up with folate that includes a methyl group, and then that methylfolate can act as a methyl donor and donate that methyl group to other transformations and biochemical reactions that are going on in the body. However, the master methyl donor isn't methylfolate, it's something called SAMe, so S-adenosyl L-methionine, which from now on I will refer to just as SAMe, but that's the master methyl donor, and we'll come to more detail on this shortly. Okay, higher level overview. So the folate cycle and the methionine cycle are 2 out of 5 cycles that are usually described in the context of methylation, but they are the most important in terms of generating SAMe, the master methyl donor. So you can see on the left there, we've got the folate cycle at a very high level, starting with folate and ending up with 5-MTHF, which is methylfolate. I'm going to go into more detail in a minute, so that's the key point for now. Then that methylfolate is needed as a cofactor in the methionine cycle for converting homocysteine to methionine using the MTR gene. So that's really the second step. It's a little bit more complicated than that as well, because it also needs methylated B12, which is a kind of mini cycle. So B12 gets demethylated by that action of MTR, and then it gets remethylated again and goes round and round its little circle. And the objective is to make methionine from homocysteine through that route. There is another route from homocysteine to methionine, which is straight up through the middle using BHMT. And then finally, methionine gets converted to SAMe using magnesium as cofactor and ATP with a gene called MAT1A as the enzyme. So then we get the target, the ultimate goal, which is SAMe. Okay, so to drill into detail, MTHFR. So I mentioned methylfolate, which is right at the bottom, 5-MTHF, and the fact it needs to be generated from folate. So this diagram gives a much more detail around the steps that are needed to convert folate to methylfolate. And what we see here, just to explain the symbols, the blue shapes are the substances, the purple are the cofactors like B3, the uppercase bold letters are the gene or enzyme names, and the little coloured dots, the ambers and the greens, are someone's personalised results. So those are the SNPs. So green means no variance, good. Amber means heterozygous, maybe some change or compromise, usually slower, but sometimes faster. And we don't have any reds on this, but red would mean even bigger impact. So There are lots of steps involved. Firstly, there is transport and absorption, so people can have SNPs on the RFC1 gene, for example, that can really impact that. DHFR is needed to convert both folic acid to DHF and DHF to THF. So if you've got a SNP on DHFR and you've been asked to take folic acid, that can be quite negative in terms of you being able to utilise that folic acid. But I'm not going to go into much more detail than that today. You could have a very big debate about folic acid versus folate. So the other steps are MTHFD1, and then finally, on the quest to methylfolate, or the route to making it, we have a gene called MTHFR, which is the one that people really focus on most. And that's because it's the only way of getting that final step done. So from 5,10-methylene THF to methylfolate, there is only one route, and that uses the MTHFR gene. So if people do have variants on there, it can have a significant effect on their ability to make that final conversion, and therefore it can have a significant effect on their methylfolate status. There are upsides though. So the fact that that is blocked can mean that there is more folate actually within the folate cycle, which can be good for DNA synthesis and repair, because there's just more of it going round to that little group of genes and substances on the left with the TYMS gene there. So some of that folate will be converted to methylfolate no matter what, but it just might be slower. So what you might think about doing if you saw a result like this, and if the person was expressing symptoms of low methylfolate like fatigue, depression, miscarriages, fertility, cardiovascular disease, there are a lot of conditions that are related to this, then you could think about testing. If you needed to actually support methylfolate, you could do that by supporting methylfolate directly through supplementation, encouraging them to consume more of the raw substances or substrates, so folate foods and cofactors. So it's not just about the folate or the folic acid, it's about making sure that the person has all the cofactors that are needed to support those biochemical changes. So that's quite often why people recommend Bs in a complex, because often one B doesn't work on its own, it needs the others to support it. Okay, so here's some examples again of what you might see in a report. So you've got a DHFR gene that looks quite good, a TYMS gene that has a variant that might impact DNA synthesis and repair. What would you do about it? It has B2 and B9 as cofactors, and so you make sure those cofactors are there. And with MTHFR, that variant, again, this is what a report might look like. It tells you what the problem might be, and it tells you, hopefully, what to do about it. Okay, so moving on to the second cycle, this is the methionine cycle, and as we said, the main objective is to make SAMe, and the secondary objective is to recycle homocysteine. So we don't want homocysteine in the body longer than it needs to be or more than it needs to be, because it's toxic. So this needs methylfolate from the previous cycle, and it needs B12, methylated B12, and it also needs zinc as a cofactor to make that little cycle on the left go round. or it can go up through the middle, through the betane route, in which case it needs betane, which can be synthesized from choline, and we need the PEMT gene to help with all of that, and the BHMT gene to actually help with the homocysteine to methionine conversion. So these are some genetic results that might be shown in this context. So MTRR, SNPs might slow the remethylation of B12. In that case, you might consider methylated B12 as a supplement, or SAMe. FUT2 we did discuss in the intro, and in this case its relevance is in connection with B12 absorption. So this person is a secretor, and so they have less good B12 absorption potentially, and potentially more risk of H. pylori infection. The PEMT gene, PEMT is actually supported by estrogen, and so something to be aware of is your client male or female? How old are they? What are their estrogen levels likely to be? Is that likely to be supporting PEMT or not? So we do sometimes find with females that this pathway has been not problematic, but then when their estrogen levels drop after menopause, then it can become compromised because they've no longer got that support. So in this case, you can think of estrogen as a cofactor for PEMT. And then finally, MAT1A is the final step for making the SAMe. And to support that, you can help with the cofactor magnesium, and make sure that they're getting sufficient protein in their diet to supply the methionine. Okay, so this is a kind of 10,000 feet view on the folate cycle and the methionine cycle, and also a little bit what happens after that down from homocysteine. So some of the symptoms of methylfolate insufficiency: birth defects, miscarriages, anemia, fatigue, cardiovascular damage, oxidative stress. Quite a lot of things just because people's methylation isn't optimal. And so here are some functional tests that might be relevant in the context of methylation. So functional tests, really, really important to look at alongside genetic testing. So you could do a B12 test. If they've not got enough B12 in the first place, then that's going to impact that cycle, the MTRR, regardless of whether they've got an MTRR SNP. But it might be worse if they have an MTRR SNP and low B12. Homocysteine. So homocysteine being too high is quite a good indicator that the whole methionine cycle isn't working very well. And as well as the homocysteine being high, that's likely to mean that SAMe is low. So that can be quite a good indicator, apart from it being damaging in its own right. But at least this can tell you maybe why the homocysteine is high. Another measure is the SAMe to SAH ratio. So you always want the SAMe to SAH to be higher SAMe. SAMe gets converted to SAH, and if SAH is high, that acts as a negative feedback loop, and it slows down the synthesis of SAMe. And then finally, ammonia. So high ammonia can be an indicator of high homocysteine, and that homocysteine actually coming down out of the bottom of the methionine cycle and generating ammonia. So high homocysteine that isn't being converted the right way to be utilised in making SAMe, but going the wrong way to making ammonia. So that's why the ammonia indicator is relevant. Okay, So support considerations, just to sum up, B complex quite often. So there are so many different B vitamins involved in these cycles that if methylation is suboptimal, often a B complex approach would be good. Depending on the SNPs, it might be more or less appropriate to look at methylated forms of those B vitamins, but it isn't always relevant. So the reason we test is because some people aren't going to respond as well to methylated versus non-methylated, and it's like everything else. We want to do our best and try and get it right first time, rather than just hoping for the best and throwing something at a problem. So this allows us to make informed decisions about what forms of nutrient to use. Other nutrients that are important are zinc and magnesium, because they're also cofactors. So we would always recommend going low and slow with supplements in case there is an adverse response. And if there isn't an adverse response, we can up the dose. So low and slow is the slogan. I've got another little result for you down there, and that's the CBS. So when I talked about ammonia being created from homocysteine, that's more likely to happen if there is a SNP on the CBS gene, which upregulates or speeds up that pathway. So the effects of that, unfortunately, is to increase ammonia and divert that homocysteine to making ammonia rather than glutathione, which is the way we would want it to go. So if someone does have a CBS SNP, then you might also look at ammonia detoxification if you've tested it or you've got other reasons to suspect it. And you might also look at glutathione or other antioxidant support. Glutathione is a much bigger picture, which is the topic of another session. Okay, these are some methyltransferase genes. So any gene whose name ends in MT, that stands for methyltransferase. And that means that they need a methyl group as a cofactor. So that's why methylation is so important. And these are just some of those genes. So on to steroid hormones, and specifically focusing on estrogen. So this is the estrogen synthesis pathway. So we start off with cholesterol that gets made into progesterone, which converts to DHEA, and testosterone. And both of those go towards making estrone and estradiol. And then the various other forms of estrogens that are needed. So the key things here in the synthesis part of the process are stress on the left. So stress upregulates cortisol production, and in doing so robs the androgen and estrogen pathway of the raw ingredients that are needed, so the substances that are needed by them. So that's why when we're looking at estrogen balance and estrogen synthesis, stress is generally viewed as being a negative thing, because it can deplete, almost before you start, it can deplete those pathways of the things they need to get started. So that's stress and cortisol. The genes involved in making estrogen are, first of all, CYP17A. So you can see that comes into play at converting progesterone to DHEA and then DHEA into testosterone. So we have an example result here at the bottom where we have a GG homozygous variant for 17A1, and that can, in this case, mean faster conversion of cholesterol to androgens. And the reason that is reported as being negative is that can mean just too high androgens. So that can happen, and it's generally viewed as a negative thing for it to be too quick and too much. The next part of the pathway involves the CYP19A1 gene, and that converts DHEA and testosterone to estrone and estradiol, respectively, and then they can convert between themselves to each other. So CYP19A1, again, if that is upregulated, it's generally seen as a negative thing because it's implicated in estrogen dominance. So we don't really want that to be working more quickly than normal. So if someone does have a SNP on that gene or the 17A1 gene, we might look at what we could do to slow it down. So you can see there in purple, they are cofactors, just like on the other diagrams, and in orange are the inhibitors. So if someone is estrogen dominant or they have SNPs on CYP19A1 in particular, then we might want to look at, are those inducers coming into play as well? So things like inflammation, insulin, and adipose tissue. So if they are, we want to try and mitigate that and avoid them. And so, help people manage inflammation, blood sugar balance, and also their weight, if weight is an issue, because they will add to the problem. On the other hand, the inhibitors are going to hopefully slow down this conversion. So we want to do that. So we want to basically use those substances to help slow down the activity of these genes. So that's green tea, DIM, which is found in cruciferous veg, and zinc. So in this case, if someone had those SNPs, you would be looking at recommending green tea, DIM, and zinc, and trying to minimise inflammation, insulin, and fat to try and balance it out. So those are the main things involved in synthesis. The next stage is activation. So this is actually converting those estrogens to active forms of estrogen. And there are really three forms that we're looking at. So there's 16-OHE1, so 16-hydroxy-estrogen, 2-hydroxy-estrogen, and 4-hydroxy-estrogen. So of those, as indicated by the colour coding, the 2-hydroxy-estrogens are positive and protective and good. So we want to encourage those to be made, and we want to encourage that pathway. Whereas the 4-hydroxy are generally associated with free radicals, DNA damage, and cancer risk. So if someone does have a predisposition to synthesizing 4-OH, we want to discourage that, try and slow it down, and try and push everything in the direction of the good protective 2-hydroxy. So you can see here that the genes involved in those are CYP1B1 is responsible for synthesizing 4-hydroxy, and SNPs on that can indicate upregulation of that pathway. So if that is the case, we would want to inhibit it, and grapefruit is a known inhibitor of that gene and that pathway. Things that are going to stimulate that pathway are stress and PAHs. So we want to mitigate the things that are going to stimulate it, and actually utilise things like grapefruit to try and slow that down. For the next pathway, the CYP1A1, that is needed to synthesise the good 2-hydroxyestrogen, if someone has variants on that gene, they are more likely to slow it down. So in this case, we're showing an amber, a heterozygous result. So we would want to try and stimulate that pathway so that the body is making more of the 2AH, and the cofactors or things that are going to help with that are DIM, so cruciferous veg again, or you can supplement as well, and ellagic acid, which is in red berries. So those are the good nutrients that are going to help someone synthesize the good protective version of estrogen. The 16 hydroxy, the 3A4 gene, the CYP3A4, is the root to this, and that's generally seen as negative as well. So we don't really want that to be encouraged. In this case, the person has a green result, which is no variance and no impact. And a bit like for the CYP1B1, SNPs on the 3A4 are likely to upregulate that pathway, which we don't need. And it's the same things that will help slow it down as for the 4OH. So grapefruit juice can be really helpful in that case. So that's the synthesis and making the active estrogens, and really trying to encourage the 2-hydroxy. Okay, so the next stages are really about metabolism and elimination. So methylation comes into play here, so that's really why we focused a lot on methylation in the lead up to this. And the gene that is responsible for methylating those hydroxy estrogens is the COMT gene. So you can see that the name of the COMT gene includes those empty letters that mean it's a methyltransferase, and therefore it needs a methyl group to work. And we've got SAMe there as a cofactor next to the COMT to show that SAMe is the methyl donor for that reaction. So if someone has SNPs on the COMT gene, that usually means that the activity of that gene is slowed down. It is further slowed down by estrogen, actually. So the more estrogen someone has, it's almost as if that estrogen is using up the enzyme in the activity of metabolizing or methylating it. And magnesium is also helpful as a cofactor in that reaction as well. So we want to encourage COMT. If someone has got SNPs on COMT, we might look at how we would support it more. And really it's about optimizing methylation as much as possible to support that pathway. And then the next phase or stage is elimination. So that's taking those methy estrogens, the 2-MeOH and the 4-MeOH, and breaking them down further so they can be eliminated. And the groups of genes involved in this are, for the methylated ones that COMT has acted upon, it's the salts and the UGTs. So there are a few salt genes that support this, and they enable sulfonation. And the support for those genes, all the cofactors, are sulfur foods. So things like cysteine, N-acetylcysteine, methionine will help. And foods like garlic and onions and leeks are also very supportive for that. If someone does have SNPs on there, then it's just more important to get those cofactors and nutrients in. The other gene is, or family of genes, there are a couple of genes called UGT that glucuronidate the estrogen in order to eliminate it. So again, if there are SNPs on here, it's really about fibre, and calcium D-glucarate is the supplement that would help support this. An indication of this might be problematic is gut dysbiosis. So yeah, so calcium D-glucarate can basically help with that as well. So that's the UGTs. And then what we haven't discussed is glutathione and the GST genes. So these are responsible for directly breaking down the 4-hydroxy, or the results of the 4-hydroxy estrogen. So free radicals that are generated by that. Glutathione is really, really important as a cofactor for the GST family of genes to help break those down and mop them up. So we did discuss glutathione in the context of methylation. So firstly, there's the need to make glutathione, and then secondly, there is the need and ability to utilise that glutathione by these GST genes, so the GSTM1 and the GSTP1 in particular. And yeah, if there are SNPs on there, again, you might need as much antioxidant support in general as possible to just really help that. Those are some genes that can actually be deleted or present as well. So it's more than just one change in the code. It's generally a bigger change, which can generally mean a bigger impact. Okay, so this is what it looks like on a report, for example. So you can see the pathways more or less the same as you've just seen on the previous slides, showing the synthesis, the 19A1 and the 17A. So in this case, the individual has got a SNP on 17A1, which is likely to upregulate, and on 19A1, which is likely to upregulate. So we'd want to try and slow that down. I'll come to that in a minute. And then activation is the CYPs, and methylation is COMT, and the actual detoxification is the SALT, the UGTs, and the GSTs. On the left there as well are the receptors. So these impact response to estrogen, and some of that can be good or bad, and the detail is in the detailed report. So there you are. That's how it would actually come out with an individual's results on there. So just to zone in on some of that, the upregulated 19A1 and 17A1 impacts higher estrogen. Of course, depending on how stressed they are and if there is DHEA available and cholesterol available to make this in the first place. But what you would want to do is encourage green tea, DIM and zinc to inhibit those pathways and try and manage inflammation, insulin and excess weight, which would upregulate those pathways potentially even more. Okay, activation. So we can see here that the CYP 1A1 is less active. So basically that's the area that we would zone in straight away. You can see there are greens, we don't need to worry about those too much, but the ambers is where the focus needs to be. So the impacts there could be less or slower synthesis of the beneficial 2-hydroxy estrogen. So we want to do everything we can to encourage that and make up for those SNPs, and that is DIM, so cruciferous vegetables, for example, and red berries will help there. Okay, and then finally the COMT. So COMT is also amber. So there's a SNP on COMT that will make it less active and slow the conversion. So we want to support methylation with B vitamins and magnesium. You might want to do a methylation test to see what aspects of methylation are more or less likely to be impacted and need support directly. And then for detoxification, this is possibly the area of most concern on the entire report because there are SNPs all over the place. So the overall impact would be to slow down that estrogen detoxification. And so we look at glutathione and antioxidant support for the GSTMs, sulfurous foods for the salts, and calcium D-glucarate for the UGT. Okay, so there you go. That's all of it in one place, summarised together with, this is a summary of the protocol recommendations that you would make for that individual. Okay, so hopefully that's helped to join the dots, and if it hasn't, here is a summary. So we haven't covered everything on here. We've focused on methylation. I think methylation is a really important starting point because it can have so many effects on so many different things. So it's a really, really good foundation to start with and to look at and to try and optimise for someone. And then we've looked at estrogen today, and it's clear that there are connections between estrogen and methylation, mainly for the COMT. That's the obvious one because that's a methyltransferase gene that needs methyl groups and needs SAMe to support it. But also the GSTs, because they use glutathione, and glutathione synthesis is dependent on methylation working well. So all these things are connected. We didn't look at detoxification in detail, but we did mention it in the context of methylation, specifically the ammonia, and a lot of the genes that are involved in estrogen detox are also responsible for breaking down and metabolizing lots of other things too, including COMT. So COMT not only detoxifies or methylates estrogen, but it's also responsible for breaking down neurotransmitters, which is really, really important, and it's a big connector between mood and feelings and hormones, for example. COMT tends to be used up or favoured or prioritised for use by the body for hormone metabolism rather than neurotransmitter metabolism. So that will take priority. So if there's high estrogen and pressure on COMT from that side, it can mean the neurotransmitter pathways get out of balance because there's not enough COMT left over for them. So that's just an indication of the interconnectedness of these different systems and the multifunctional nature of some of the genes and also the interconnectedness between them, and why it's important to view genetics in context. So in context of each other, in context of cofactors, and in context of the person. So right to go back to the beginning, what symptoms are they presenting with? What life stage are they at? What is their diet like? What is their environment like? And how is that all interacting with the genes? And what functional tests might you want or need to do to support the hypothesis that you have based on genetic results? It's just all has to be looked at as part of the bigger picture. Okay, so that's the end of the technical part of the presentation, and this is just a summary of some of the reports that Lifecode does, some of the tests, and also to say that we do run a lot of practitioner training events and seminars, and this is one of those events that we have scheduled for the summer, 9th of June, and it's all about female health, genetics and functional testing. So if you sign up for that, you can get a half-price Dutch complete test and a half-price Lifecode estrogen balance test. and you can put all the pieces together, and we'll help you make the most of those results and learn how to get the best for your clients at that event. So it'd be fabulous to see you there. And if you're a BANT member, you get £20 off. And this is some information about the practitioner support that we have. So we provide telephone support for practitioners for every order that they've made, if they want it. And you can contact us on the details given there. Thank you very much.